Hello everyone, in this video we're going to talk about some similarities and differences between DNA and RNA as well as the properties of each. So we're going to start with DNA. DNA is a type of macromolecule called nucleic acid. So DNA is one type of the macromolecule nucleic acid, and it controls all the actions in the cell. So you can kind of think of DNA as the computer code that directs how everything is done on your computer, but in the end is not really the DNA that is shown, right? It's not the computer code that you see on your computer, but it's the code that directs how everything is done. So DNA is ultimately the instruction on how everything is done in the cell. Its primary function is to store genetic information. So genetic information means we have genes, and genes are a part of DNA, and that allows one cell to become two cells, how the cell is supposed to be working, how a multicellular organism is supposed to be built. So all this information is coded in the DNA. And DNA stands for deoxyribonucleic acid. As you can see, this word contains the letter D, N, and A, which is why we call DNA DNA. But really, its full name is deoxyribonucleic acid. So you need to know this word. But in order to remember this word, you kind of need to break it down a little bit. So DE generally means you don't have something. So DE means you're missing something. Oxy should remind you of oxygen, right? So DNA, deoxyribonucleic acid, is actually missing an oxygen in comparison to RNA. Ribonucleic acid, so ribo is the sugar, or deoxyribose is the sugar that's in DNA, and because DNA is a nucleic acid, the N and the A should make sense to you. The difference between DNA and genes. So we talk about DNA a lot, we talk about genes a lot, but how are they related to each other? So you can think of DNA as one large piece. It's quite long of a molecule. But genes are parts of the DNA. So one DNA can contain a lot of different genes. So it says a gene is a sequence of DNA that codes for a protein or a trait. We say it's a sequence of DNA because we have a long DNA, but this one long DNA doesn't just have one gene. Otherwise, you would only have not a whole lot going on in your cells. So this one piece of DNA has tons of different types of genes, and those genes can code for either a protein that can do something in your cells or a particular trait for you as an organism. In humans, genes vary in size from a few hundred DNA bases to more than 2 million bases, right? So if you think of a long piece of DNA, there are chunks of it that codes for different things, and the amount of bases, DNA bases, that it contains, or the number of monomers of DNA is different depending on what the gene is, right? So some genes could be this long. Well, as an example, some genes are longer than other genes, and then that's how it is. The Human Genome Project, so when we think about a human, just for your information, you don't actually have to remember these numbers, but a human had between 20,000 and 25,000 genes, right? If you think about it, you should already know that in each eukaryotic, in each human cell, right, human somatic cells, you should have 23 pairs of chromosomes. If you only have one gene on each chromosome, you only have 46 genes. But that's not how it works. We have 20,000 to 25,000 genes, which tells you that each piece of DNA has a lot of genes on it. And then it says most genes are the same in all people, right? If you think about it, in the end, although we have a lot of differences, and you can see the differences when you look at a person, when you look at two different people, you can tell that we are still people, right? I have ears. Whoever's watching this video hopefully still have ears, right? I have a nose, you have a nose. In the end, our bodies are built in a very similar way, right? That's what makes us human. So besides the difference, most of our genes are exactly the same, which makes us human. Moving on, when we talk about eukaryotic DNA versus prokaryotic DNA, there are a few things that you should notice. For prokaryotic DNA, you just have one piece of circular DNA, right? It's still double-stranded DNA, it's still a twisted ladder or a double helix of the DNA, but it's a circular shape. A eukaryotic DNA is linear, but besides the shape of the DNA, we also have something called histone proteins added to the eukaryotic chromosome. So within the eukaryotic genome, we have DNA wrapping around the histone proteins. So this helps organizing the DNA, right? If you think about it, the eukaryotic DNA should have way more information than a prokaryotic DNA does, and you have this long piece of DNA. How do you keep it organized? You wrap it around some histone proteins, which helps keeping everything organized. What else? Something else that happens is that if you think about it, if you have DNA wrapping around histones really tightly, this part of the DNA cannot be expressed, meaning that proteins cannot come in and do transcription and translation and actually make, well, you can't actually make proteins out of this piece of DNA if the DNA is wrapping around the histone proteins too tightly. But then if you have other parts of the DNA that's not wrapped around as tightly, then this part of the DNA can actually be expressed and you can actually make products out of it, right? So why is this important for us to know? If you think about your body, you have all different kinds of cells, but in the end, most of your somatic cells are exactly the same in terms of the DNA that it carries, but they're very different from each other, right? Your brain cells are very different from your red blood cells. What enables that difference? One of the ways that the difference can happen is that the wrapping pattern, which part of the DNA is wrapped really tightly, you can't produce anything out of it, versus which part is wrapped not as tightly, you can produce products out of it, is different between your brain cells and your red blood cells, okay? When we think about organizing DNA, you can remember when you talk about the cell cycle, when DNA is replicated and then eventually split, when the cell is divided, we actually wrap the DNA around the histone so tightly that it becomes an X shape, so that it's much easier to split the two DNA pieces apart, right? If you think about it, if you have two headphones in your backpack, for whatever reason, the ones with wires, and you don't wrap it, then the two headphones are going to get tangled pretty easily, right? Same idea with DNA. You have all this DNA. If you don't keep it organized in some way, all the DNA are going to get tangled. Okay, so now we're moving on to DNA structure. You should know that a DNA is stored in the nucleus of a eukaryotic cell. By this point, you should know that a prokaryotic cell does not have a nucleus, so DNA cannot be stored in the nucleus for a prokaryotic cell, right? That doesn't work. And DNA, again, is a type of nucleic acid and it's made out of nucleotides. So you can think of nucleotides as individual Lego pieces, and a nucleic acid is almost like a Lego house, right? You're building this Lego house based on some sort of instruction and how you recombine the pieces. So you have the nucleotides, you put it in a chain, a certain type of sequence, eventually that gives you a long piece of DNA, and you can call that a type of nucleic acid. Each nucleotide, so each Lego piece, right? If you think about a Lego piece, you have the parts that is bumpy, and then you have the other side that is not bumpy, right? It has a certain shape, a certain structure, which gives it a certain function, right? Same idea with DNA. It has a structure which gives it the function of being able to be used as genetic material. So when you think about a nucleotide, there are three parts to it: a sugar, and the specific type of sugar in DNA is called deoxyribose. And then we have phosphate. My apologies, my dog is barking. Phosphate, a phosphate group, just like ATP has a phosphate group, DNA also has a phosphate group. And then we have nitrogenous base. There are 4 different types of nitrogenous bases in the DNA as well. So the 4 types of bases is what makes different nucleotides different from each other. The 4 bases are A, T, C, and G. Okay? So moving on, when we talk about, again, DNA has different bases, right? So let's take a look at this picture and take a look at the 3 parts of a DNA. When you look at a DNA, we have the 5-carbon sugar, we have the phosphate group, and we have the base. So the sugar and the phosphate are always connected to each other on the one strand, right? Again, DNA is double-stranded. On one strand of DNA, you have sugar, phosphate, sugar, phosphate, sugar, phosphate. That's what keeps the one strand together. But DNA is double-stranded, so there has to be something that keeps the 2 strands combined to each other, right? So that's what we call DNA base pairing. So the base pairing is when you have the C base and the G base, those 2 can form 3 hydrogen bonds in between each other. And then when you have the A base and the T base, those 2 can base pair and form 2 hydrogen bonds in between each other. The C and the T simply cannot match, A and G cannot match, and so on, okay? And again, this picture right here shows you one nucleotide. It's one sugar, which is a 5-carbon sugar. It's a pentagon, okay? That's always the sugar. There's one base, or one phosphate on the outside, and then we have one base. And we call this the sugar-phosphate backbone, right? If you look at this picture, we have the sugar-phosphate backbone, and then we have the base pairing in the middle. We call it the sugar-phosphate backbone because it's on the outside, is what is the backbone, right? Okay. So the base pairing, again, we can also call it a complementary base pair because they're not exactly matching, but they sort of match in a way, right? You need to remember that it's called a complementary base pair. So A always pairs with T, or the reverse. T always pairs with A, C pairs with G, G pairs with C. DNA is a double-stranded molecule. It looks like a ladder with two sides. It's like a ladder with two sides, and then you twist like this, okay? DNA, nucleotide base pairing. We already talked about that. There's also something called the 5-to-3 direction. Or we can say that the two DNA strands are antiparallel to each other. So what do we mean by that? If you look at this picture right here, you can see all the pointy part of the pentagon is pointing up on the left side. And all the pointy part is pointing down on the right side, right? It's not going like this. They are parallel, but the direction of the parallel is opposite, okay? So how does that really work? What do we mean by 3 and 5? The 3 and 5 are really counting sugars, right? So I'm going to explain what that means, but you don't actually need to know it for the test. I'm simply explaining it in case you're curious, you really want to know what 5 and 3 are about. So if you look at this oxygen and we count clockwise away from the oxygen, you can count 1, 2, 3. So this is 3. And then you count 4, and 5. So 3 is pointing down on the left side, and 5 is pointing up, right? You can see on the picture too. It says 3 is over here and 5 is over here. On the opposite side, we have oxygen as well, and you count clockwise 1, 2, 3. Now 3 is pointing up. 4 and 5. 5 is now pointing down, right? So again, we're saying that the two strands of DNA are in opposite direction. It's 5-to-3. If it's 5-to-3 going down on the left side, then it's 5-to-3 going up on the right side. More on DNA structure. This double-stranded molecule is coiled, rotated in a helical manner, right? Because it doesn't just match, and it's not just anti-parallel, it also twists. So we say it's a double helix, right? And again, the backbones are made out of alternating sugar and phosphate molecule. And okay, look at this one more time. So we have sugar, phosphate, sugar, phosphate, sugar, phosphate on the outside, and then inside the DNA where we actually pair the two strands, that's where we have the A pairing with T on the inside, A pairing with T and C pairing with G. But on the outside, the backbone of each strand, we have sugar, phosphate, alternating from each other. Okay, this also shows you again the 3 and the 5, and then on the right side, it should be 3 over here, up, and 5 going down. Moving on. Sorry, my dog is bothering the rabbit. Hey, come here, dog. All right. So, and again, this picture shows you one more time the base pairing is in the middle, right? Complementary base pair, A pairing with T, C pairing with G, and then the 3 parts we have sugar, deoxyribose, which is this 5-carbon sugar is always a pentagon, and then the phosphate is the little one on the outside, and then the one in the middle, the bigger one, is the nitrogenous base. So now you can take a look at these questions. Well, let's see. What are the building blocks of a DNA? It's a nucleotide, right? Write the complementary sequence. A, the first one, pairs with T, C pairs with G, and you keep going. And if it tells you the direction is 5-to-3 to the left, on the bottom, the 5 will then be over here, and 3 will be over here. And the arrow should be pointing to the right. The other type of nucleic acid, right? We said DNA is one type of nucleic acid. The other type is called RNA, ribonucleic acid. So the only difference is the deoxy. And again, as I mentioned earlier, DNA has one less oxygen actually on the sugar compared to the RNA. So RNA, what it does is that it makes, well, there are different types of RNA, but the specific type of RNA that we'll focus the most on is called messenger RNA, and messenger RNA makes proteins and allow gene expression. So what we mean by that is we can't go from DNA to protein directly. There has to be an intermediate called RNA. So RNA helps with gene expression. You go from the DNA code, you have to transcribe the code into an RNA code, and then the RNA information is then used to help you figure out how the amino acid is supposed to be connected to each other to make the protein. Okay? And again, you see over here DNA replication, transcription is DNA to RNA, RNA to protein is called translation. There are 3 differences between DNA and RNA that you need to know about. RNA is generally single-stranded. That doesn't mean that all the RNA are single-stranded, but all the DNA are double-stranded. So we're saying that DNA is double-stranded and generally RNAs are single-stranded. And the mRNA is definitely single-stranded. And then we have the sugar in the backbone of an RNA is a ribose, and the sugar in the backbone of a DNA is deoxyribose. And in terms of bases, DNA has A, T, C, and G. RNA just simply doesn't have a thymine. It has A, U, C, and G, okay? So how does that work? When you have A, it has to pair with U, right? If you're going through transcription, you have this DNA strand, let's say you have ATT, the first A, you're trying to make RNA, okay? So the DNA is the A, you're trying to make RNA from this DNA code, using this DNA code, which means the U, instead of pairing A with T, you'll pair A with U. And the next one, T can still pair with A, and the next one, T can pair with the A again. So you can take a look at this Venn diagram and see DNA and RNA both have A, C, and G, but there are 3 differences. Double-stranded, single-stranded, deoxyribose, ribose, uracil versus thymine, okay? If you understand this meme, that is probably 1 question on the test. Have a wonderful day. Let me move on to the next video.